This is the second video on control of gene expression in bacteria. We'll continue to look at the lac operon in this video. So last time we looked at how the lac operon functions, and we looked at the individual effect of a mutation in each of the different parts of the lac operon. And we looked at how that mutation would affect the production of either the beta-galactosidase gene from the LAC-Z gene or the permease gene from the LAC-Y gene. So just to review this a bit, in a normal situation, and if you have a wild type operon, you have repression of both the Z and the Y genes when you do not have the inducer lactose. So you're lacking lactose in the environment, and the LAC-I repressor will bind onto the operator and repress transcription of those structural genes. I hope you guys figured out that when you have a mutation in the LAC-I gene itself, or if you have a mutation in the binding site for that LAC repressor, which would be the LAC-O, that you cannot repress those structural genes as you would expect, and both genes would now be turned on even when lactose is absent in the cell. There's a term I'd like to introduce regarding these mutations, and that is the term constitutive. So the term constitutive is used to indicate that a gene is always on. It cannot be repressed as it should be. Because these mutations are constitutive, we actually call the operator mutation an OC mutation. So C stands for constitutive. So next, I'd like to tell you about some additional experiments that Jakob and Monod did on this lac operon to figure out how it worked. So what they did is they used something called an F' plasmid to create a cell, a bacterial cell that had two copies of the lac operon. The way that F' plasmids are created is actually shown here, and we don't need to know the details, but just suffice it to say that bacterial cells normally just have one of every single gene. They are not diploid like we are, but if you put an extra copy of that lac operon on a plasmid, a small extra piece of DNA within the cell, you can create a situation where you have two copies of your gene of interest. So these cells would be partial diploids for the lac operon. So next, I'd like to move to the worksheet and look at numbers 7 through 10. So on these numbers 7 through 10, we list two copies of the lac operon. And so you can see the first copy of the lac operon has all of the parts. It has the LAC-I, the repressor, the promoter, LAC-P, LAC-O, which is the operator, and then the two structural genes we're investigating, the Z gene and the Y gene. And the two copies of the lac operon are separated by this dash. And so here's one copy of the operon, and here's the second copy. The other thing I'd like to note is that these are all in one cell, and what we're really interested in is the overall effect of having both of those in the same cell, of whether we are making the beta-galactosidase protein in the absence of lactose and then in the presence of lactose, and also for the permease gene. So what I recommend doing for these types of problems is to start off by looking at the structural genes that we're interested in investigating. So the Z gene and the Y gene in this case. Here you can see that there is a functioning Z gene, a Z plus, on that first copy of the operon, but a mutant Y gene. On the second copy of the lac operon, you have a non-functional Z gene, but a functioning Y gene. And so one way to think about this is that you're going to need to transcribe this copy of the operon in order to get the messenger RNA for the Z gene and to produce beta-galactosidase, and you will need to transcribe this copy in order to get the messenger RNA for permease and the permease protein being produced. So these are all in one cell, and all of the regulation in this case for both of the copies of the operon is wild type. And so this should just function as wild type. So for the Z gene, in the absence of lactose, the repressor would bind to the operator and transcription would be turned off. And when we added lactose, then the repressor would respond to that lactose by changing its conformation, falling off of the DNA, and allowing this copy of the operon to be transcribed, and therefore we would make the beta-galactosidase protein. Same type of thing for the Y gene. So the normal regulation is that the LAC-I, the repressor, turns it off in the absence of lactose, and when we add lactose, it makes that repressor fall off of the DNA, and now you can transcribe the LAC-Y gene to get permease. For the second one, this is a little bit more complicated. So here you have two wild type genes on that first copy of the LAC operon, so the Z and the Y plus were on the first copy, and the second copy has a mutation in both the Z and the Y genes. Also, this first copy has a mutation in the LAC repressor. And from our previous work, we would expect that if we have a mutation in the LAC repressor, that you won't be able to turn off the operon in the absence of lactose, and that those genes would now be expressed constitutively. However, that's actually not what happens in this case. When you have two copies of the LAC operon, you can actually get the functioning LAC repressor that is transcribed from the second copy of the operon. And since this is a protein, it can actually go and bind to any functioning operator sequence in the cell. And that could be either that operator sequence on the same LAC operon that it was produced from, or it could also bind over here to the operator on that first LAC operon. And so the result of this is that you get this functional LAC-I, and it can actually go over and repress the transcription of that operon in the absence of lactose. And so when you don't have lactose, you just get the normal situation, which is the operon is turned off. And when you do have lactose, then lactose binds to that repressor, it changes conformation, and it falls off, and then you can transcribe and get both of those proteins being produced. So here we have two copies of the LAC operon, and you have a mutation in one of those LAC-I genes. The other one is functioning. Because the LAC-I is actually a protein that is encoded on that gene, that protein no longer is tethered to the same copy of the operon from which it was encoded. And so you can make that functioning protein, and it can go and it can diffuse around the cell, and it can bind to any functioning LAC-O sequence that is found in the cell. And so this one here will kind of substitute for the absence of the LAC-I from the other copy of the operon. The term for this is that the LAC repressor will act in trans. It can act on either copy of the LAC operon. Next, let's look at number 9 and number 10. So in number 9, we have the same situation that we have the wild type Z and the Y gene in the first copy of the operon, and then a mutant Z and Y gene in the second copy of the operon. In addition, we also have a mutant promoter in that first LAC operon, while the second promoter is wild type. So, in order to get the functioning Z and Y genes being produced, we need to transcribe this first copy of the operon, and we cannot do that because we have a mutant promoter. Now, the question that arises is whether this functioning promoter over on the other copy can actually substitute for the mutant promoter on the first copy. And the answer to that is no. And the reason why that promoter can't substitute is because the promoter is actually a DNA sequence, and that DNA sequence is physically attached right next to the genes that it regulates. And so this particular promoter can only regulate these particular copies of the Z and the Y gene, just like this promoter can only regulate these copies of the Z and the Y gene. And so in this case, there's no way that we can ever turn on expression of the Z and the Y genes because we have a mutant promoter. We can't get RNA polymerase bound to that promoter, and we can't transcribe. And so across the board, those should be negative. The last example has two functioning Z and Y genes in the first copy of the operon again, and two mutant copies in the second copy of the operon. The first copy of the operon also has an OC mutation, that is a inactivating mutation in the operator such that the repressor can no longer bind to it. And so from what we learned before about the OC mutations, we would expect that to make the Z and the Y genes constitutively active. This is indeed what we find, and that is because this functioning operator in the second copy cannot actually substitute for this mutant operator in the first copy of the operon. Again, the operator is a DNA sequence, and it only regulates the genes that are right next to it on the operon. So it would only regulate these particular genes, whereas this copy of the operator would only regulate those genes that are right physically next to it. The result of this is that you can't bring in the repressor to turn this off in the absence of lactose, and so the Z and the Y genes would always be on in all cases. And there's a term for how the promoter and the operator act. They act in cis. And so things that act in cis will only affect the genes that are right next to it on the chromosome. And so here's an example of that. You're making the repressor, and you have a mutation in the operator sequence that mutation makes this particular Z gene to be constitutive. It's always on. And even though you have a functional LAC-O over on the other copy of the lac operon, it cannot substitute for this mutant copy because it's a DNA sequence and it's only regulating those genes that are right next to it on the chromosome. There's one additional type of mutation that Jakob and Monod identified, and that is something that we call a LAC-I super repressor, and we refer to that as a superscript S. So this particular mutation was pretty interesting because it's a mutation in the LAC-I repressor, but it's a mutation that affects only part of the function of the LAC-I. The thing that it can still do is that mutant LAC-I superrepressor can still bind to the DNA, but it cannot actually respond to lactose. So lactose comes along, and normally the repressor changes its conformation and falls off of the DNA. And this superrepressor now can't respond to lactose, and it just stays on that operator. And in fact, there's very little that can be done to actually get that repressor off of the operator once it binds to that operator. So I'd just like to point out that the LAC-I superrepressor mutants also can act in trans. And again, the reason why that is the case is because this is a protein. Even though it is encoded on one of the copies of the lac operon, it is no longer tied to that particular location where the gene was encoded. once it becomes a protein, and that protein can diffuse through the cell and bind to any functioning operator sequence. To end up, I'd like to discuss two complex situations. So, first example is if you had two things that give opposite phenotypes, which one of those would actually determine the phenotype? And so, say you had a situation where you had a lac-I super repressor, so that one should bind to the operator and completely repress all transcription. But you also had an OC mutation in which the repressor could not bind to the operator, and therefore the genes on that copy of the operon would be constitutive. So in this case, the one that wins out is actually the OC. And the reason is because that mutant super repressor cannot bind in the first place to that mutant operator. Second example is if you have a P minus mutation together with an OC mutation, which one would win out there? So normally an OC mutation, you can't repress it, and now those genes get transcribed all the time. But if you also have a P minus mutation, you can't get RNA polymerase to the DNA, and you can't actually transcribe. And there's really nothing that can win out over a mutation in the promoter. And so if you have both of those, the promoter mutation would win out, and you would not transcribe the genes of that operon.